In this series, we'll speak with scientists from the Dangoor Center for Personalized Medicine to gain an up-close understanding of what happens in their research laboratories, which developments we can expect to reach the market, and what medicine will look like in the coming years. This time, we're hosting Professor Yaron Shav-Tal. Professor Yaron Shav-Tal is from the Goldman Faculty of Life Sciences and the head of the Dangoor Center for Personalized Medicine. And he recently began his tenure as Vice President for Research at Bar-Ilan. Mazal tov, congratulations.
Thank you.
Professor Shav-Tal's field of research is gene expression. In his lab, they track RNA activity and other particles within the cell in real time while the cell is still alive, using advanced microscopic technologies and fluorescent molecule labeling. Professor Shav-Tal completed his doctorate at the Weizmann Institute and his postdoc fellowship at the Albert Einstein College of Medicine in New York. Since 2005, he's been conducting research at the Goodman Faculty of Life Sciences at Bar-Ilan University, and he's received numerous awards and research grants, including the prestigious grants from the European Research Council, or the ERC, and the National Institutes of Health from the United States. Before being appointed Vice President for Research, he served as Vice Dean of the Faculty of Life Sciences for a decade, and he followed that by 2 years as Dean. So, Professor Shav-Tal, welcome. Thank you for joining us.
Thank you. That sounded very familiar.
Oh, that's good, because it was your bio.
Yes.
So if it was unfamiliar, then we'd be in the wrong place here, right?
Exactly,
So
yeah.
At least we started with something easy,
Yes.
right? So you study the cell, the actual cell. You look under a microscope and look and see what's happening. Tell me a
Right.
little bit about the process. What do you see? What are you looking for?
Well, first of all, you have to realize that cells are transparent. They don't have any color, except for our red blood cells, right? But most cells, you just look at them and you don't see much. You can see the body of the cell, which is called the cytoplasm. And if you have keen eyes, you can also see the nucleus, which is where all the DNA is inside. But other than that, you don't really see too much. So in order to understand what's going on within the cell, we have to label, we have to color some of the molecules that we want to see, want to observe, and then follow them under a microscope.
So how do you do this? This is the fluorescent protein process. I know the terminology.
Yes.
I don't have a clue what it means. So tell me about the process.
Exactly, yes. So, well, for many years, people could color or paint molecules in the cell, but you had to kill the cell first. You had to fix the cell in
When you say paint, you don't mean paint.
Well, it's almost like painting because we work, in the lab and we do things with our hands. So it's,
Really?
it's
You
almost
don't
like...
just sort of put in dye and it's attracted
Oh
to
yeah,
certain
you
areas?
put it in, but you can imagine it as work being done. You can't see the molecule, so you're not painting the molecule, but obviously it gets done when you put the fluorescent molecules or the color molecules onto the cells. So as I was saying, so for many years people could color different molecules or proteins within the cell, but you had to fix the cell first. What we try to do is to follow this in living cells. So in order to follow a molecule in a living cell, you have to somehow label the molecule before you go to image it, and you have to keep the cell alive. So this is what you mentioned beforehand, the use of fluorescent molecules or fluorescent proteins, which was a technique that was developed some years ago, I would say a few decades ago, using a molecule that comes from a sea creature, Medusa.
Medusa, which
Yeah.
is jellyfish.
Jellyfish, exactly, yes.
Right.
Jellyfish.
I love the term Medusa because it gives you such an image, right, with the hair.
With the hair and everything,
tentacles.
exactly, yeah. So a jellyfish, and some of the jellyfish, if you go to an aquarium, right, you see that they fluoresce in different colors.
They're beautiful.
Green, blue, red. So people were studying this in the US and trying to understand what this molecule is in the jellyfish, just for the sake of understanding, because it's interesting. We don't have any fluorescent molecules in our bodies, and most organisms don't, but these sea creatures do have it. And therefore it was interesting. And they found this molecule which they termed or called green fluorescent protein, GFP.
Is it always green?
Well, it's not always green. If you wanted to fluoresce, which means if you wanted to send out a green halo, you have to shine onto it blue light. So in the jellyfish, there are cells that generate blue light, and the blue light in these cells in the jellyfish go to other cells in the jellyfish, and it turns bright green.
Fascinating. So how does that map onto what you do?
Okay.
in terms of the cell under the let's start even more basic.
Yeah.
How do you keep a cell alive? Where are you getting these cells from? What's the process here? I
Okay.
want to understand.
So cells can come from many sources. Most of us tend to use cancer cells because they grow forever. So you can keep them in the lab, proliferating, growing, dividing.
That's the problem with cancer cells.
Right, exactly. So that's an advantage for science, but a disadvantage, obviously, for the patient who has a tumor. And also, it gives us the ability to study a certain cell in my lab in Israel and study the same cell in a lab in Germany and in the US and in Japan, because we're all using the same cancer cell that was developed many, many years ago. You could also take cells from a creature and grow them in the plate as well, but then you would encounter a problem that cells tend to die. They senesce over time, and that's why we use cancer cells, because they don't die. They die, but they don't die. They always have a progeny.
Right, as you say, offspring.
Yes,
but
exactly.
Same thing, same idea here. So the actual cell, you take cells, you put it under your microscope, you paint them?
Well, first you grow them in a plate, a special plate, and you give them an environment which makes them happy, which means that they have sugars, and they have fats, and they have CO2, and they have O2, so everything that they would need to grow. And if you're using cells from a human body, you also give them the right temperature, 37 degrees, like in our body. And if they're happy, so then you can see under the microscope that they are spread out and they look happy. Okay, they look satisfied.
Joy,
yes,
right?
they're satisfied. Yeah. And then you put them under the microscope, and microscopes are not these little things that you might imagine,
What I had in
third grade.
Grade lab.
Microscopes can be, the size of this table, even larger. They cost a lot of money, and they're very state of the art usually. And in order to see things in the cells which are at high resolution, you have to use an objective that can come really close to the cell, and therefore we use very powerful objectives to see what's going on in the cell. As I said, the cells are transparent, you don't see much, but if we paint the molecules in the cells, I guess we'll get to this in a moment, so then you can actually see what's going on in the cell in real time. So going back to the green fluorescent protein, which was discovered in a jellyfish, and generally, what do you care about it? It doesn't mean anything to us. But then somebody had this bright idea, and they got a Nobel Prize for this bright idea. Okay, there were 3 people who got the Nobel Prize for the discovery and the use of the green fluorescent protein. The first guy who discovered the protein, the second guy who had the bright idea to say, a protein is a protein. So what does it matter if I take a protein from a jellyfish and I stick it onto a protein from a mouse? I fuse them together, and now I'll have protein X from the mouse with the GFP on it. Let's see what happens. And that's what he did. And lo and behold, this is what we do until today. Well, the third guy, maybe I'll just finish with the second guy. So now what we do is we take any protein of interest in any organism, doesn't matter which one, as long as it has proteins, we fuse the GFP protein to this protein. So now we have GFPX, and we express it. We send it into the cell, and now the cell has this molecule called GFPX. The third guy who got the Nobel Prize, he had a different approach to looking at proteins. He wasn't so interested in fusing them or anything like that. He wanted to generate new proteins. So let's say the jellyfish has an original protein which fluoresces in green. He said, what would happen if I would take this protein and mutate little pieces inside it? Would it change the characteristics of this protein? And what he discovered was that the answer is yes. And suddenly he had a red fluorescent protein, and a yellow fluorescent protein, and a blue fluorescent protein. So that's why I was saying painting, because now we can take different fluorescent proteins, express them in the cell at the same time, and then you can see different molecules in all the colors of the rainbow, basically, at the same time.
It sounds like it completely, it changed the field, transformed the field now.
Completely changed the field.
So much that's happening there.
Right.
What do you see when you look under a microscope now? Because you study the cell.
Right.
What are you looking for?
So it depends what you're looking for.
What are you looking for?
Well, we're looking for RNA, so maybe that's the next step. We'll get to that in a moment. But I'll just say that when you look under the microscope under these conditions, everything looks black, because when we use fluorescence, we want the light to be the only light that is coming out. We don't want any other light interference. So everything is black. You don't see anything. You just have to believe the cell is there. And then depending on what you labeled, that's what you will see. For example, if I want to see the DNA in the cell, the nucleus of the cell, so I'll take a molecule that is originally situated inside the nucleus of the cell, and you'll see nice, very round areas in all the cells. This is the nucleus. If you want to see the cytoplasm, you would take a molecule which sits in the cytoplasm and color it to a certain color. If you want to see mitochondria, that's the energy plant of the cell that makes all the energy, the ATP. So you would label an mitochondrial protein, and you would see mitochondria very beautifully in the cell. So anything that you want to see, you can see in the cell, but you can't see too many proteins, different proteins at the same time, because then it would just be a big mess.
Right. Sounds like it would be beautiful, but apparently
it's very
not
beautiful.
helpful.
Yeah. So
all the colors of the rainbow simultaneously.
yes, it's very beautiful. The idea, though, is not just to make cute pictures, which is nice.
Do you have some on your wall?
on your wall, on the calendar.
Right, exactly.
Yeah,
You can make a calendar of cells.
Some of the companies do that.
Really?
So we have.
Hopefully not cancer, because that's not really something happy you want on your wall.
But you have very pretty and elegant pictures, but we want to learn something from that.
Of course.
So doing this work in a live cell means that you can actually see what's happening in real time. And then you can understand how things really work. Because a lot of the other type of science that people do, for example, we said beforehand, we fix the cells, we kill the cells at a certain point. So now you have a snapshot of this exact time when the cells were fixed. But you have no idea what happened 5 minutes beforehand.
You can't see the process.
You don't see the timeline. Yeah.
Right.
And definitely if you do biochemistry, so there's no time there as such in a specific cell, because you take many cells, you quetch them, you take everything out, and then you look at what was there in these cells, usually in a million of cells at the same time. When you do live cell imaging, you're looking at single cells, and you can see the difference between single cells over time.
That's fascinating. So you said you study the RNA.
Right.
Is RNA a protein? Because it's made up of nucleic acids, not amino acids, which is what I was always taught. Proteins were amino acids.
Right, so it's confusing, right. So proteins are amino acids, as you said, and exactly DNA and RNA are nucleic acids. So these are two different molecules altogether. They both have acids on the end of their name, but it doesn't mean anything to us at the moment. So amino acids are the building blocks, the Lego blocks that build the proteins.
Right.
And RNA will be built from Duplo blocks. And Duplo and Lego, they don't fit together.
That's right.
Okay.
But you could use this fluorescent protein, the same process to paint the nucleic acids.
So that's exactly the problem, because if you want to take now GFP, which is a protein,
right,
and stick it onto RNA, it just doesn't
click, like you said, the Lego and
so there's just two different animals, they don't go together. So somebody had to come up with some kind of bright idea.
Really?
And in the lab where I did my postdoc at the time at Einstein, as you mentioned, that's where they generated this trick. They said, okay, we can't attach GFP to RNA directly, but we can do this indirectly. We can take a protein, which is a protein that has GFP on it, and get it to bind a specific RNA. And then you have the RNA, let's imagine it like a little string. And on the end of this RNA, we attach a few binding sites. And now these fluorescent proteins will bind there in tandem, a whole series of them. And now one molecule of RNA has this little light on it, made of many tens, actually, of GFP proteins that are bound to it. And actually, RNA is not a little
it's not a straight line like this?
No, it's a ball. Okay, so now you see these
so
little...
it's a ball wrapped in fluorescent...
Exactly. Wow. So you can see
it's
these...
Like Christmas.
It's like Christmas. It's exactly like that. So you can see these little guys traveling in the cell, and they look like little balls. And now you can follow their life cycle, which wasn't something that one could do beforehand. So this was really a breakthrough. So GFP and all the fluorescent molecules that I mentioned, they were a huge breakthrough. They changed the way people do biology, not only in cells, even in organisms. You can have a fluorescent cat and a fluorescent fish if you want. You can purchase them if it's really something.
Cat?
Yeah, if you
so
want.
My cat could glow in the dark?
Yeah, if you think that's something that you need in your house, that's definitely
a night light.
Exactly. You just have to You have to have the blue light onto the green
That's right.
to get a green
Okay.
cat. You have to remember that. But some people buy fish, that
Yeah,
have it
fish
in
see.
aquariums.
Right.
Yeah, but people use this for science as well. So they use fluorescent proteins in animals as well. Going back to the RNA, for RNA it's more difficult to do these type of experiments. So this is what we are unique in my specific lab. There are other labs over the world that can do this, but this is something that you really have to invest energy in order to get to observe the RNA in living cells.
So I'm going to go back to my question.
Yes.
What are you studying? It sounds like basic science. And I don't mean, sometimes people hear basic science and I think it's dismissive.
Yes.
Basic science is a specific terminology that means like studying the way things work, not necessarily addressing a specific problem, not necessarily trying to fix them, but really understanding at its basic level
Right.
what's going on. Are you doing basic science and
Yes.
really just exploring? Because it's the foundation for so many other things,
Exactly.
right?
That's the base, that's the foundation, and our flag is basic science. If you don't understand basic science, you can't go on to develop therapies, medicines,
Right.
new techniques. You have to understand how things work. It's like a car, you have to understand how the car works if you want to generate a new type of car, if you want to fix it, right? So same thing with understanding basic science in biology. So we are interested in RNA, and in order to understand how an RNA functions in the cell and what happens to it over time, you have to use the techniques that we are using and to ask the questions that we are asking. So what we try to do is to understand the life cycle of the RNA from the time that it is born. We call this transcription.
Okay.
In a cell. So you have a gene, a DNA, and this DNA generates RNA, so it transcribes RNA. So obviously just this word transcription is something that keeps hundreds of labs very, very busy for decades, okay, just to understand what's going on in this little moment of transcription. And after that, you have this RNA that has to move from the DNA, which is in the nucleus, to the cytoplasm where all the action is happening. So the RNA is the messenger. The DNA doesn't move, so it's protected in the nucleus. We have to keep our genome protected that nothing bad happens to it, but the message can travel. It's like a photocopying machine. Okay, so the DNA is photocopied, made into an RNA, and this RNA now has to travel. But now if we kind of zoom into what's going on in the cell, so in the nucleus, there's a lot of DNA. It's like mountains of DNA and oceans of molecules
You're such
moving
amazing
around.
metaphors. I'm just picturing this whole world
That's-
of.
That's the way I see things, because
Right.
I try to envisage, what the
Right.
RNA is going through. And as we said, it's a little ball,
a little
but
Christmas ball once
it's
split
actually
up,
it's
right?
not so little when you think in the terms of the nucleus. So it's like a little elephant. I usually tell my students. So this elephant is trying to move its way along, and it has these walls of DNA on each side, and it has to find its way at some point, and we would hope quickly enough, into the little exits that are found in the envelope of the nucleus. These are called nuclear pores, little tunnels or channels where the RNA must move through. So it just moves around, and one of the questions that we asked years ago was already, is there any mechanism that pushes the RNA to these holes, or is the RNA just sort of fumbling around until it finds its way? So you would think, that there's directed movement, you go there, and it's time for you to get out, you have nothing to do here in the nucleus, run to the cytoplasm. But what we found was that, and this is basic science, that the RNA just moves around by random movement, diffusion, and it could take it quite a while until it reaches the cytoplasm.
And so different RNA escape or, move forward at different rates?
Yeah, depending on their size. So smaller ones will get out more easily, the larger
They
ones
slip through.
Exactly, and the larger ones, they kind of
Lumber along like an elephant, right?
a mammoth maybe. Yeah.
A woolly tooth mammoth now.
Yeah.
I've got that.
Yeah.
The Christmas ball in the shape of a woolly tooth mammoth.
Yeah.
So, kind of lumbering along till it makes its way out.
Exactly.
To get fascinating. So tell me a moment where, doing basic science, which is obviously really exciting to you, you're not going to find a cure for cancer in your lab. But tell me a moment where you went, oh my God, I can't believe look what we just found out.
Okay, that's something that we're proud of. When I started my lab in 2005, so it's already 19 years, almost 20 years. So the question that was the highlight of the lab, which we thought at the time that we want to solve, is how fast does the RNA get out of the nucleus, but at the nuclear pore?
Mm-hmm.
So we said it's moving along quite slowly until it reaches the nuclear pore, but no one until that time had managed to detect an RNA moving through. And it was unclear why. Because basically you see the RNAs in the nucleus, you see them moving around, you see them in the cytoplasm moving around, but this moment of escape, exactly, from the nucleus to the cytoplasm, just something that nobody could see.
Really?
So it wasn't clear why. What was the reason for that? Is it too slow? Is it too fast? Is it something changing in the RNA so you can't detect it anymore? Perhaps it's throwing off all its fluorescent molecules. It's kind of shedding them when it moves. No idea. So we decided to tackle that problem. We did not know at the time that several other labs were trying to do the same thing at the same time. And luckily for us, we were the first ones to actually see this in real time and to discover. I didn't believe this in the beginning, so this is the moment that you were referring to. So obviously I had a student who was doing this, excellent student, Amir, and he came back and he said, I think I caught this. I caught this, under the camera. Yeah, and I said, I don't know, I have to see it. So you look at it, and everything is happening so fast that you're not sure if your eyes are playing tricks on you or if this is something that we actually are seeing. So it took us some time until we verified that indeed we can see numerous events like these, which are not easy to catch. And we found that the movement through the nuclear pore is less than half a second, 500 milliseconds. We couldn't say exactly the precise time, but if the movement from the gene to the pore is, let's say, 30 minutes, so the movement through the nuclear pore of this big thing, this big structure, is less than half a second. So it's as if something is kicking this football out through the channel into the cytoplasm. So we were the first to publish this, and this was very exciting for us. And a few months later, some other papers came out as well.
They weren't first.
They weren't first, but we have to acknowledge that they did some things better than us. And they could actually say that the time that the RNA spends in the nuclear pore altogether is even less than what we measured. Not 500 milliseconds, half a second, but 200 milliseconds, which is even shorter. And they could break down even, the attachment to the nuclear pore, the movement through the nuclear pore, and the release on the other side. So they could break this down. So this is something that
is fascinating. Is there something that kicks out? Why does that happen so quickly? If it's fumbling around, and then why?
Well, you'll be amazed that there's some things that are still not understood
in science.
In science,
so
in basic science.
Have stuff to do.
We still have so much stuff to do.
Good. Next interview, you'll come back and explain it to me.
I was always worried before I became an investigator with my own lab, if I would have original ideas to study. I thought, I looked to the left, to the right, I saw the people with careers, many questions for many years. What would I ask for, for 30 years? Well, let me tell you, there's no lack of questions. There's a lack of people to do the work.
Right, I get it. So this is, I find it fascinating. But let's talk about the people who are not such science geeks like you, like me. What are the potential implications, consequences? Why should the average person care? What does this matter? Okay.
That's a legitimate question. It's not always easy to answer this and to explain it, but I can say that after COVID, it's very easy to explain.
Of course, all the RNA. Yeah, go ahead.
Suddenly people became aware that there's a molecule called RNA.
That's
and
right.
now when I have to explain to people what RNA is, so I don't have to...
I know that already.
Everybody knows already what RNA
We're
is.
all experts in it, that's right.
Right, because the SARS-2 COVID virus is an RNA virus, and the vaccination that was used is actually an RNA.
Right.
Well, most of the vaccinations, not all, but most of them, the ones that we got here in Israel and in the US, were RNA vaccines. So RNA is a very unstable molecule. When I was a student at the Weizmann Institute, almost nobody worked on RNA.
Everybody was looking at DNA. Yeah,
DNA, proteins,
right.
RNA, not so interesting. But there was one person in the lab who worked on RNA, and she had this cabinet which she used to lock with a key, and you weren't allowed to breathe next to the cabinet. Why? Because RNA is so unstable, because there are all kinds of enzymes that like to attach to the RNA and eat it up, degrade it. So the belief was, if you just look at the RNA, it's going to disappear. So we know today that indeed RNA is very unstable. Obviously, it occurs in the cells, but maybe connecting to what I said beforehand about the life cycle of the RNA. So the RNA also has a point in which it disappears from the cell. It dies off or gets degraded, as we say. So RNA is all the time undergoing generation and degradation. So it's like a continuous cycle. So RNA is very unstable. So if you want to send RNA into a person and to actually have it reach its target, which are the cells inside after you vaccinate, and it takes quite a while until the RNA, the vaccine, reaches its place, and to remain stable and to get translated into a protein that would be a protein which would imitate the virus, this takes a long time. So how do you keep an RNA alive in the body, outside cells, in cells, for such a long time? So if you would have asked me this question 10 years ago, even 5 years ago, the answer would be you couldn't. But for some reason, miraculous events, the time at which this virus attacked us was the time that this basic science matured into the ability to generate a vaccine, which is an RNA, and this RNA would be able to be stable in the cells for quite a long time.
But then it goes away.
And then it goes away, obviously.
Well, the reason I'm saying that is because all the kind of negativity around the vaccine was how it was going to transform your body. I think people are confusing DNA and RNA and understanding that RNA has a life cycle. And in some ways that's challenging, and in some ways that's good. Right. Because, I was talking to a scientist once about, I think at Bar-Ilan, about mutating or cutting RNA. Yeah. And if you mess up, it's not great, but it goes away. Exactly. DNA is a little bit of a problem. Yeah. So it's actually really a fascinating molecule that you're exploring.
Right. I mean, that's the way the cell thinks of RNA. It doesn't want to generate a molecule that's going to be stuck there for 20 hours or even for 20 days. It wants a molecule that will be there for half an hour, 1 hour, sometimes even several minutes. So you might ask, so why go to all the effort of generating it? It takes a lot of energy. You have to pay, the cell has to pay a lot of money to generate the RNA. It's not such a great idea. But the cell wants to act like a thermostat, it wants to feel what's going on all the time. If I need this molecule, I'll make it. If I don't need it, I'll get rid of it. And if 2 minutes later I need this molecule again, I'll make it again. So the cell wants to be very flexible. It doesn't really stuck in one situation that it cannot move left and right. So that's why the RNA, in its generation, in its being, is a molecule which is unstable, because you want to have this instability to keep the flexibility. But going back to the vaccine, so many people studied basic science of RNA over the years, and they discovered different properties of the RNA, which they then tried to modify. Okay, so we have an RNA that is made out of nucleic acids, and the cell recognizes them and degrades them very quickly. The question was, can we make some kind of block in the nucleic acid, a Lego block, that would be a Lego block that would allow this RNA to survive for longer. Obviously, for sending this into the cell, it's of no use. So it's not a good idea. But if you want to work with this in the lab, that would be a good idea. And then people generated these new elements in the RNA, which made the RNA stable. So now I have a stable RNA. I can use it as a vaccine to send it into the cells, and it will remain there for a reasonable amount of time until you get whatever you need from this RNA, and then it will disappear. So you've lengthened the life of the RNA, and now you can use it as a tool in all kinds of things, but also as a vaccine. So that's how basic science led to the understanding of how an RNA normally acts. And now I can say, what can I do to change it, to improve it, to modify it, whatever.
So it's all the basic scientists who saved the world from COVID.
In the end, yes, because some of these scientists, people didn't believe them in the beginning.
I know.
They thought they, Meshugaim.
Right. You can't do it. It doesn't make sense. And the whole, right now, again, we're all well versed in RNA. We're all experts, but nobody had heard of it. Nobody knew what it was or thought that it was relevant. And it was all the basic scientists who were committed to exploring and understanding. I said it sort of facetiously, but it really is true.
Yeah.
Basic scientists save the world.
Yes.
You're the head of the Dangoor Center for Personalized Medicine. What's personalized about what you do?
What we do? Well, I'm not sure if what we do is exactly personalized because we're trying to understand the general things. But in our research, we do come down at certain points to some things that are more applicable, perhaps, or connected to disease. Obviously, it's always interesting to take the basic science and then to work on something which is connected to some kind of disease. For example, we work now on different stresses that occur in cells and how they affect the biology of RNA in the cell. It was found years ago that chemotherapy, which is a stress to the cell, causes the formation of certain structures in the cell, and these structures can hide RNA inside them. And if the cell survives the chemotherapy, so then these structures dissipate and the RNA is now ready to go. If the cell is killed by the chemotherapy, so we don't care. But cells have ways to evade the chemotherapy. So we're trying to understand how these structures, these granules, we call them, in the cells, stress granules, that's the name, because they occur under stress, they hide the RNA inside them. What can we do to perhaps increase the generation of these granules under certain conditions? And perhaps this will make the cells more susceptible to the chemotherapy or less susceptible, depending on what you do. We weren't really sure what we would find when we set out to do this. And we found that we can give different treatments with the chemotherapy that would make the cells more prone to death. So you can use these properties that we have now understood about RNA and the proteins that bind to RNA in order to take this another step further and say, this is what chemotherapy does to the cells. I can make the chemotherapy more potent if I give now another hormone together with the chemotherapy, and now the cells will die more rapidly.
Too easy.
So we're not only staying in the basic level, we try whenever we can to move to the more applicable milieu of experimental science. But obviously we always remember what we set out to do.
Right.
But it's always interesting to look at a disease, to understand something. We have a student in the lab who's understanding how herpes viruses infect cells. It's interesting because the virus takes over the cell and uses all the machinery in the cell for the sake of the virus. So it shuts down all the cellular work that's going on and takes this cell hostage, basically. And now the cell is like a slave. It's making all the molecules for the virus. The virus builds up as a virus, explodes the cell, kills the cell on the way. And now you have more viruses. So we're trying to understand how the nucleus is changing with RNA is made, how the nucleus is changing under viral infection.
Every time I talk to a scientist who's doing basic science, meaning, exploring the body, I am blown away by how complicated, complex, fascinating, amazing the body. I mean, we're just talking about a cell. How many cells do we have in our body? Millions?
Trillions.
Trillions, right? And you're just talking about, and even within that, it's just, it blows me away by how extraordinary this thing that we live in, I mean, it's amazing. Basic science.
That's great.
Incredible.
I think it's a wonderful job that I have. I get to sit down and look through an eyepiece into a cell. I'm only looking at what interests me. Obviously, the tens of thousands of different molecules that I haven't even looked at because it's not my area of expertise, and there are other people who are doing that.
30 years, if you run out of things,
exactly.
you were worried. You got plenty to do.
Yeah.
How did you end up focusing on this? What made you choose the RNA molecule? Especially, you said there was only one woman who locked everything up.
Yeah.
How did you decide, I'm going to get the key to that cabinet?
Well, during my PhD, I worked on proteins. And the proteins were connected to RNA. They bound RNA. But it was a field that was only developing at the time. And actually, the lab that I was working in didn't work on this. Luckily, my professor, he was very open. So he let me do whatever I wanted, basically, in this little field. And then when I looked for a postdoc, I was looking for a place where I could follow proteins in cells, and I came upon this lab where they were looking at RNA. So I thought, wow, RNA, it's really interesting. So I know about proteins, and proteins that bind RNA, so let's go and work on RNA. And that's why I went to this lab where I did my postdoc and learned how to tag RNAs in living cells and to play around with RNA and proteins in cells. It was a great time. And when I came back to my lab, I decided this is what I'm going to be doing for the rest of my career.
Which is, but was there something about the RNA that was like that it had a lifespan, that it was somehow unique? Was there something about it?
I think at the time it was because, as I said beforehand, detecting RNA in a cell, in a living cell, was impossible. Up until that point, I was lucky to come to the lab at the moment where this technique was developed. So this was
like eureka. Oh
exactly.
It's a whole new world.
Yeah, it's a whole new world. There's so much to do.
One of the pioneers.
Yes, exactly.
Got it. And amazing where we've landed. You said 19 years later, right? Yeah. You've been with your lab. You just took on the role as head of the Dangoor Center for Personalized Medicine. Yeah. Tell us a little bit about the center. Tell us. I mean, I do all these interviews with different investigators who are associated, but you have like so we say Rosh Gadol, the big head, the big picture. Tell us a little bit about the center. What's your vision for it? Where is it going? Where is it now?
First of all, I'll tell you that it's a beautiful initiative that was started some years ago, and thanks to the Dangoor family. And it's a way to bring science to the public. And if you go on the website, I didn't know too much about it, so I sat and I looked through the different movies and different articles. And you're interviewing me now, but people have been there beforehand on the website, and it's really amazing. I also see what people are looking for on the internet, because we get the reports, and I see that the information on the website is going to so many people who are looking for basic information about science, medicine, biology, and they find it on the website. So in the Dangoor website, you can see there are over 100 researchers, women and men, all from Bar-Ilan, and they all connect somehow to personalized medicine. Some people connect better, some people less, but basically it's everybody is connected in a unique way. We have people from biology, from medicine, obviously, but we also have people from psychology, because today we want to tailor specific treatments, psychological treatments, especially in this time now, to people who have undergone, whatever they've undergone, specifically tailor it to them. And the website and the information there brings a lot of new information and current information to people who are searching to understand what personalized medicine means.
Which is really the future of medicine, right?
Which is the future. I think we're already living the future, okay? We have already today medicines that are tailored to certain people, just by the fact, even today in certain cancers, they take the cells from the person, they sequence the DNA in the cells, and they can say, the doctors in the hospital, they can say which mutations this specific person has in their specific DNA. Let's say breast cancer. So there are different types of breast cancer, or melanoma. There are many types of melanoma. The treatment would be the right treatment to the mutation that you have, to the defect that you have in your DNA. It's not a very bright idea, and this is what has been done for decades, to take the same chemotherapy and bombard anybody who has melanoma with the same chemotherapy. Obviously, it's not going to work on some people. It will work on other people. So if you can have the information prior to the treatment, in order to tailor the treatment to the specific person according to what's written in their DNA, you have a better chance of curing this person. And this is what people are doing today. So this is happening already. We have also treatments that are, it's almost science fiction, that are tailored to specific genetic diseases that are very rare. And they have a specific mutation in their DNA which causes this terrible disease. And there's no way to cure these people, because the mutation is built in all the cells of their body. But now, using personalized medicine, you can tailor the specific mutation which is causing the defect and correct it in a way.
Amazing.
And now these people that they have a chance.
Right.
You're not going to cure them, obviously, from the disease altogether, but you will give them a better chance to have better quality of life, prolonged life. The world is changing in that respect. And even now with the vaccines of the RNA that we spoke about, in the context of COVID, what the companies were doing at the time, before COVID came around, they were thinking something different altogether. They weren't focused on any virus. It just happened to be that they had the technology, and that was immediately shifted to produce the vaccine. But these companies were generating vaccines for cancer. Mm-hmm. So instead of using chemotherapy, you're going to get an injection with this RNA or whatever RNA, and this is going to cure your cancer. Okay, so this.
How far along are we with that?
That's very far along.
Okay.
Yeah, they were already far along beforehand. They just had to.
So we're getting there.
We're getting there. Definitely, it's amazing.
It is. Just to look into the future. So tell me, 10 years, your vision for your lab, for the field of RNA. Mm-hmm. Where are we in 10 years?
My dream.
Let's start with that. That's always a good place.
My dream is that we would be able to take our science that we do in cells, cancer cells that we grow in a little plate under the microscope, into the animal, into the organism. And it's not so easy to do. People are doing this already in certain places, but you need really huge microscopes, very high-tech, very state-of-the-art, very expensive. Not everybody can do this. And integrate the full organism, that you can look inside an animal and see what's going on inside. That's already something else. Wow. And we already have this going on in different places in the world. Mostly people will do this, I would say, in flies or fish or things like that.
Not an elephant.
Not an elephant.
Because you got elephants in my head. I'm trying to see a mobilized elephant under a microscope.
No, but in mice.
In mice. Okay, we got mammals.
Start with mice, yeah. So that would be interesting. And if we could do that in my lab at some point, I would be very happy. So that's the dream. That's where we're going. And I think also what's beautiful about science and about this job that I do as a scientist is that you really don't know what's going to be in a few years' time, or even tomorrow. Right. A student comes with some discovery, something that you hadn't thought about beforehand, and says, "Look, I saw something interesting. Do you think it's interesting?" So usually we have to be open-minded enough and not dogmatic to say to the student, go ahead, try it and let's see. And that's where the really interesting and breakthroughs come from these little moments of somebody paying attention to something that nobody would notice otherwise.
Right. The mark of a brilliant scientist is curiosity.
Curiosity. Definitely.
And openness and just wonder.
Yeah.
I mean, I listen to you and I love your metaphors. I love the way you view. No, because people can look at it as a tiny little, but it's not. It's a world.
It's a world. Yeah.
A world on its own. And the things that you're doing are pushing us all forward, not just in terms of knowledge, but really for those who aren't as curious, let's say, has real consequences and real meaning. And our world is a better place because of your little world, or big world that's miniature.
Exactly.
So thank you for everything you're doing. Thank you for coming in and talking with me today. It was fascinating.
Thank you. Thank you for asking all the right questions.
I'm sure there are other ones somebody else would have asked that would have been right also.
Okay, next time.
Next time. I said we're going to bring you in and we're going to be able to talk about where you're at in the future. So thank you so much, really. This was a joy and a pleasure.
Thanks.